Can somebody confirm that you can hear me?
We can hear you.
Great. Sure, we're beyond that. All right, before we begin, are there any questions? I have done some grading. I'm currently on, I think it's lab 4. Let me close this down. Let me take a look here. Sure. Last week, I have not graded the extra credit study group 4 yet, but I got lecture 4B, or the worksheet for lecture 4 graded. Actually, I'm working on worksheet lab 4 graded, and I'm working on worksheet lab 5. I'm almost two-thirds of the way finished with lab 5. I hope to have it finished by tonight, in which case, well, if I get it finished by tomorrow morning, um, I will then require that the second grading of it will be this Saturday. If I do not get it done graded by Wednesday morning, then the second grading will be later. Let me state that Lab 5, a lot of students are getting some questions wrong. Um, hopefully you're not seeing the screen here. Let me make sure. Yep. If you don't understand a question, you should ask. If you're not online, you can always email me. But especially questions 5 and 6, a lot of students have gotten them wrong. I haven't graded 8, 9, and 10 yet, or 8, 9 yet, but um, most students are graded questions 1 through 7. There's a few who I haven't graded 4, 5, no, 5, 6, and 7. I'm working on those last few. I'll get that done shortly after the lab tonight. But I'm not sure about getting the entire thing done. It looks like I've only got questions 8, which shouldn't take me that long. And then question 9, that shouldn't take me long either. So hopefully I'll get it done by Wednesday morning, in which case the second grading will be due this Saturday. And a lot of you are going to want to do a second grading because you've not done very well on this lab. If you have questions, like I said, you should ask and I will help you. All right. That's why I make myself available in the lab. Let's go back to the get out of the grade book. Get back to the assignment. I gotta get to the Zoom and share my screen again. All right. I have that we had talked about group translocation, a special form of active transport seen only in prokaryote cells, where it's similar to active transport in that something is transported either into the cell or out of the cell. It does require energy, but that energy may not come from ATP, like it comes from a high energy phosphate compound like PEP for group translocation. What's really different is that the molecule is chemically changed when it is brought either into or out of the cell by the active transport protein, like glucose being moved into the cell will be changed if it's using group translocation to glucose 6-phosphate. Any questions about group translocation or active transport? Remember, in eukaryotes only have active transport. All right, so let's talk about the cytoplasm. The cytoplasm is the substance inside the cell membrane. So actually, inside the cell membrane is everywhere in a prokaryote cell. The cytoplasm is mostly water, about 80% water. It does have dissolved solutes in it. And in the cytoplasm can also be the dissolved DNA. The nucleosoid is the region where the DNA happens to be floating in the cytoplasm. That region tends to look different, but there's no boundary between the cytoplasm and the nucleoid region because it's wherever the DNA floats in the cytoplasm. Ribosomes and inclusions are found within the cytoplasm, but realize that the ribosomes and the inclusions are not dissolved in the cytoplasm. They are too big to dissolve in the cytoplasm. Just a minute here. I'm sitting on something. Don't know why I didn't notice that before. All right. Any questions about the cytoplasm? Let's get rid of that. The nuclear area, called the nucleoid region in a prokaryote cell, is where the DNA, usually a single chromosome, is located. There is no nuclear membrane, so the nuclear region, or the nucleoid, is a part of the cytoplasm of a prokaryote cell. Now realize that's very different from a eukaryote, and they do have a nuclear membrane which divides the cytoplasm from the nucleus. You can find in the cytoplasm in some prokaryote cells a plasmid. We'll talk a little bit more about that later. A plasmid is a small molecule of DNA. It differs from the chromosome mainly in its size. The chromosome is very big, very many genes on it. The plasmid is small with just usually a few genes on it. Ribosomes are something that we find in the cytoplasm, but ribosomes are way too big to be dissolved in the liquid or the water of the cytoplasm. So they're not dissolved, they're sort of like a precipitate. And there you can see the artist's rendition of them. Let me blow this up here. Each of these dots, so you can see there's lots of them in the cytoplasm, is a ribosome. Ribosomes are the site of protein um, manufacture in the cell. Any question about ribosomes? Let me state that prokaryote ribosomes are smaller than eukaryote ribosomes. The prokaryote ribosome is 70S in size. That S is the sedimentation coefficient where the ribosome sediments on an ultracentrifuge gradient. And that's the way biochemists and molecular biologists define ribosomes by their size and that's how they sediment, where they sediment in the ultracentrifuge gradient. The ribosomes of a eukaryote is a little larger. It is 80S in size. Ribosomes are made up of both protein, so you can call it ribosomal protein, and ribosomal RNA. Both of them together make the ribosome. Any question about any of that? You don't need to know that the ribosome is made up of subunits, but there is a larger subunit and a smaller subunit. They get together to make the ribosome. An inclusion is another part that you can find in the cytoplasm, but the inclusion is not dissolved in the cytoplasm. An inclusion is a reserve deposit that the cell is storing, and it's built up so much of it that the inclusion precipitates out of solution. So the cell could use them when resources are low. So initially, when the cell sees a molecule out of the cell, it will bring it into the cell if it wants it inside the cell, if it wants to use it. And initially, while that molecule is low in concentration inside the cytoplasm, it will be dissolved in the cytoplasm. But as more and more molecules are brought into the cell, they can precipitate out and form an inclusion. And that's all an inclusion is, a reserve deposit of material the cell is keeping or making. And it becomes so high in numbers that the deposit actually precipitates out of solution from the cytoplasm. And so you can see that, like right there is an inclusion, right there. I don't know if I have another picture, probably not. When you look at the cell, I got another one coming up. Here's some inclusions there, these dark things there, the dark things here, and the bluish things there. There are different types of inclusions. You do not need to know the names or what they are. You just need to know there are different types of inclusions. Any question about any of that? So those are magnetosomes, inclusions in this cell. This is a cell that can metabolize sulfur. And so whenever it sees a sulfur molecule, it will bring it into the cell and store it as a reserve deposit. And so much sulfur is built up in the cell that it starts precipitating out of solution. And then this is a magnetosome. Let me blow this up. And the yellow line is a cell, and you can see three inclusions of the, excuse me, metachromatic granules is the inclusion here. Magnetostomes are there. You don't need to know the types of inclusions, but different cells will have different inclusions depending on what the cell is bringing into the cell or making inside the cell, and then storing it, and then it precipitates out of solution, making the inclusion. Like most cells do not have sulfur granules in them, only sulfur metabolizing cells will have them. Any questions about inclusions? Another structure you can find inside the cell is an endospore. An endospore is a specialized resting cell produced by certain species of bacteria, mainly members of the family Bacillus and Clostridium, that allow the species to survive in harsh environmental conditions, meaning an endospore can survive harsh environmental conditions that will kill the parent cell, the cell that made the endospore. So endospores are resistant to desiccation, heat, chemicals, radiation. They've had endospores surviving in the desert of Egypt for over 8,000 years. The longest living, I think, was 40 thousand years, but I could be wrong. It might be 40 million years, something like that. And they dug um, into the Antarctic ice sheath, and they were looking at different samples, and they went back something like 40,000 years, and they found an endospore frozen in the ice in Antarctica. And they thawed it out and got the endospore to germinate. So that's the oldest living endospore that we know. And I know it's at least 40,000 years. I should probably do a search of that to make sure it's not 40 million years, but um, oh, this was even older. This isn't coming from the ice of Antarctica. It's coming from a bacterial spore trapped in a crystal in New Mexico, probably a salt crystal, and it germinated after 250 million years. About that's probably something new. So it was 40 million years was the one I was doing. So let's go ahead and change that because that's something new. Endospore does is allow the cell to survive. And when you make the endospore, it's called sporulation or sporogenesis, the endospore formation. And then when the environment changes, the endospore, shown here, can actually germinate and return to a vegetative cell, which is, let me blow that up. There's a vegetative cell, which is a normal rod or bacilli and has an endospore inside of it. So remove the endospore, and that's the vegetative cell. So that will germinate into that, and that's called germination. Any question about any of that? So when the environment starts deteriorating, the parent cell will realize that it's going to die as the environment deteriorates further. And so the parent cell begins building the endospore. You can see all of them, almost all of them. No, not those. Most of them are building endospores there. And there's a close-up of an endospore. And there's the parent cell here. It's making the endospore. Let me blow this one up. All right, endospore formation happens when the cell realizes that the environment is deteriorating. It will go through cell division, but unlike normal cell division, which normally split the cell into two, it'll split into a smaller segment and a larger segment. And of course, the DNA, before the cell begins to split, the DNA has to divide. One molecule of DNA will move to one side of the cell, the other will move to the other side of the cell. And then the cell will begin to split in much the normal way, where the cytoplasm starts to divide and it starts to form, and the cell membrane starts to form. But there will be a smaller cell and a larger cell. Then the plasma membrane will separate into two cells, and the smaller one will develop into the endospore. The cell will actually get, at least this one, two membranes around it. So two plasma membranes around the smaller cell. And you'll note that that moved inside right there. And that's why we call this the parent cell. And then the cell wall will form in between the two membranes. So we have a peptidoglycan layer in between the two membranes. And then the spore coat will form. That's really thick outside the endospore. And then the environment will become so detrimental that the parent cell will die. And when it dies, it'll release the endospore. This shows you a little bit of a, oh, come on, endospore formation, but it's not terribly detailed, not very good. It just shows you the endospore forming within a cell.
As I'm in the midst of applying for residency right now, wish me luck, Grammarly has again been a lifesaver with my application and helped.
Skip the commercial. This cell's going to form there. It starts forming the endospore. Any questions about any of that? All right. Oops, went too far. So that's it for the structures of prokaryote cells. Let's move on to the structures of eukaryote cells. By structure, I mean both a membrane-bound organelle and a non-membrane-bound organelle. And also by structure, we do mean the cytoplasm, which is not really either a membrane-bound organelle or a non-membrane bound organelle. I suppose you could call it in a eukaryote, you could call it well, I'm not sure what you'd call it because it is membrane bound. It does have a cytoplasm, which makes the division. So anyways, this is showing you the structures we'll talk about with the plant cell. On the right, we're seeing the structures we can find in an animal cell. It's not shown, but the flagellum can be found in a plant cell, like fern spore cells, which is the life cycle of the fern. Um, they do have flagella, which can then swim around. I think there might be a sperm, which of the flagella also has a flagellum, but we're talking about fern. I'm not sure about any other plant cells, meaning like an angiosperm or a gymnosperm. I'm not familiar with any of those that have a flagellum, but there are some plant cells that have a flagellum, so we could put that over there. And this is showing you a peroxisome in a plant cell. You can find those. Oh, there it is there in an animal cell. Many of them can be, many of the cell structures can be found in both a plant cell and an animal cell, like a nucleus is found in both. The nucleolus is found in both, which that's a cell structure, not a membrane-bound organelle. The mitochondria can be found in both. The chloroplast can only be found in green plant cells. So a cell that is green can have a chloroplast in it. Would a root to the corn have a chloroplast in it? It's a yes or no question, people. A root cell to the corn plant, would it have a chloroplast in it?
No,
Pretty easy to answer it. I actually heard the answer is no. Does the root cells ever see sunlight? They do
no,
not.
they don't.
So a root plant, or any root actually to any plant, will not have uh, chloroplasts in it. Assuming the root is underground and it's not green, I suppose there could be a plant. I'm not familiar with any off the top of my head where, well, actually with a potato. There's one that I know where the root, if it gets out of the ground, meaning on top of the ground, so sunlight can touch it, the root of a potato, that region that's exposed to sunlight, can turn green and actually have chloroplast in it and then undergo photosynthesis. And whenever you see a green potato, that's what's happened, meaning the tuber of a potato. The where it's green was where the potato was above ground, and it is green because of chloroplast in it. But usually the root is not under sunlight and does not have chloroplast in it. And off the top of my head, the potato is the only one I know of where the root can turn green, but there are others where it probably could. Just don't know them off the top of my head.
What about an orchid where their roots are, some of them are exposed?
An orchid is a plant which is a lives on a tree, so the roots to that, actually it's not a true root, but it is exposed to sunlight. It could be green. I don't know off the top of my head if an orchid undergoes photosynthesis or not. You'd have to look it up. And if it does undergo photosynthesis, then the roots could be green and they could have chloroplasts in them. But as far as I know, an orchid is only the flower and that's not green. But obviously, let me think about it. I've seen an orchid plant. Yeah, it has green stems. Um,
I was thinking just, the household ones where you put them in the bark and some of the roots end up
are exposed.
initially.
Yeah, they're not green. Those roots are not green, so they don't have um, chloroplasts in them.
Okay, thank you.
Yeah. Anyways, the mitochondrion can be found in both. Um, there is one which is found only in the animal cells. It's a centrosome. It's a cell structure, meaning a non-membrane bound organelle. It's involved in mitosis and cellular division, which is, I guess, mitosis. But the plant cell does undergo mitosis, but it doesn't have the centrosome, which is centriole, I guess, is the specific cell structure or non-membrane bound organelle that animal cells have and plant cells lack them. They both have the Golgi apparatus. This is showing you the lysosome in the animal cell, but actually plant cells do have lysosomes, but they're much more common in an animal cell. One membrane-bound organelle that you can find in plant cells, you do not find in animal cells, is the central vacuole. It says the vacuole here, but that's actually the central vacuole. I know because the central vacuole is the largest membrane-bound organelle in the plant cells, larger than the nucleus. In the animal cell, the largest membrane-bound organelle is the nucleus. But in the plant cell, it's actually the central vacuole. Animal cells do have small vacuoles, but they're closer to the size of a perioxysome in the animal cell. And plant cells, besides the central vacuole, which is this huge vacuole, which is actually the majority of the cytoplasm in most plant cells. Plant cells can also have small vacuoles, which will be the size of a perioxysome. And we'll talk a little bit more about those. Any question about any of this? You'll note some differences in the shape and structure of a plant cell and an animal cell. An animal cell tends to be roundish, can take on essentially any shape, and that's because the animal cell only has a cell membrane. The plant cell, on the other hand, has a cell wall, and so it will be a distinct shape. This plant cell might be octagon. It's hard to see if that's 3 or 4. It might be a hexagon too if that's only 3. And then you can have plant cells which are rectangular, meaning they can be square shaped. There are some plant cells which are roundish and they have a cell wall which is round. So that's one structure you find in plant cells, do not find in animal cells. This is an actual electron microscopic image of a plant cell on the left and an animal cell on the right. And right there, they're showing you a little vacuole in the plant cell. This huge thing right there, which isn't labeled, and it has something else inside of it, but most of this right there is probably the central vacuole. I know that because the nucleus is smaller than the central vacuole, and that looks like that's probably the nucleus right there of the plant cell. Seeing the mitochondria very well there. There it is there. Oh, here it's better here. Let me see if I can blow that up. No, that's not much better. Maybe there. Oh, that's an algal cell. That's what I was saying. I was wondering why it was a funny shape because that almost looks roundish. It's an algae. Not quite a plant cell. It's close to a plant cell. All right, telling you some differences. This is what this slide is showing you. Let me shrink that down. Some differences between the prokaryote and the eukaryote cells. Difference in their cell size. The prokaryote is about 1/10 the size of a eukaryote cell. And obviously the eukaryote cell can be much bigger than 100 micrometers in diameter. The largest prokaryote cell, not, excuse me, eukaryote cell that all of you have seen is the yolk of a chicken egg, which is what? That's close to an inch in size, so close to 2.4 centimeters. The nucleus, there's no nucleus in a prokaryote cell. There is in a eukaryote cell. So that's actually one of the main distinctions between the prokaryote and the eukaryote. Membrane-bound organelles, this is the other main distinction between a prokaryote and eukaryote. The membrane-bound organelles are absent in a prokaryote cell. They are present in the eukaryote cell. Membrane-bound organelle is a organelle surrounded by a membrane, such as the Golgi apparatus. Flagella, they're a little more simpler in the prokaryote cell than in the eukaryote cell. The glycocalyx can be present in both the eukaryote and the prokaryote. In the prokaryote, if we can see them under the light microscope, we call it a capsule. If you cannot see it under the light microscope, and you have to see it under an electron microscope, we call it a slime layer. The glycocalyx is present in eukaryote cells only if they lack a cell wall. So really only a certain protists and animal cells have a glycocalyx in eukaryote cells. The cell wall is present usually in prokaryotes, but not all prokaryotes have a cell wall. And then not all eukaryotes have a cell wall. Plants and fungi have a cell wall. Many protists have a cell wall. Animal cells and some protists lack a cell wall. The cell wall in a eukaryote, if it's present, is chemically simple. Like green plants have a cell wall made up of mostly cellulose, whereas the cell wall of a prokaryote is more complex. Even the gram-positive cell, which is mainly peptidoglycan, which is in most cell walls, but mainly peptidoglycan in the gram-positive cell, there are other molecules in the cell wall. The plasma membrane is simpler in the prokaryote cell. It's more complex in the eukaryote cell. Animal cells have sterols in their cell membrane. Sorry, I said cell wall, cell membrane. Animal cells have sterols, which is cholesterol, in the cell membrane to give the cell membrane strength and support. Eukaryotes also have a lot of carbohydrates and protein in their cell membrane. Prokaryotes typically have less or no carbohydrates in their cell membrane. They do have proteins in their cell membrane. They also have transport proteins to pump molecules into and out of the cell. The cytoplasm, this says no cytoskeleton. This is out of date, and the author has never changed this table. There is a primitive cytoskeleton that's now been known for at least 15 years in prokaryotes. The cytoskeleton is a network of proteins running throughout the cytoplasm, making sort of like the backbone of the cell. And prokaryotes do have a simple cytoskeleton. The eukaryotes have a very complex cytoskeleton. They also have cytoplasmic streaming. We will show you that in a little bit. Ribosomes of the prokaryotes are smaller at 70S, and they're larger in a eukaryote at 80S. Any question about any of that? Okay, let me shrink that down. If a prokaryote can swim or move, it does so by a flagellum. That's the only structure that a prokaryote cell has for moving. Eukaryotes, some of the eukaryote cells can move by a flagellum, like the sperm cell is one that has a flagellum. Eukaryote cells can also move by cilia, like the paramecium. The trachea in our throat also have cilia. That's to move the mucus up the throat, where it'll either be swallowed or spit out. Sorry about it tickling in my throat. And then the third way that a eukaryote cell can move is by pseudopods. Any question about any of that? The flagella is a few on the cell, and they're very long. The cilia are very, very many on the cell, and they're very short. Usually the cilia beat in unison, so the paramecium, if it's moving this way, the cilia will beat that way. And usually the paramecium just moves in the direction that the cilia are propelling it forward. So the cilia are actually moving this way, the opposite way the cell moves. Anyways, the flagella and the cilia, as well as the pseudopods, are used for locomotion on eukaryotes, but the prokaryote only have a flagellum or flagella if they're plural. I already mentioned that the cell wall can be present on eukaryotes. Most eukaryotes do have a cell wall. The difference is animal cells and some protists lack them. The cell wall, if it's present, it's simple. No peptidoglycan in the cell wall. And we're only going to talk about the cell wall of plants. And it's composed of cellulose. All right, let's talk about the cell membrane. The cell membrane of eukaryotes is actually similar to bacteria, meaning eukaryotes, when they evolved, discovered that the cell membrane of bacteria was pretty useful, and so they borrowed it. And so the cell membrane is very similar between a eukaryote and a prokaryote, meaning it's mostly made up of phospholipids. It does have proteins in it, like transport proteins. The cell membrane is a phospholipid bilayer. Does contain proteins, both peripheral proteins, integral proteins. Peripheral proteins are a protein that's on one side of the membrane or the other, or at least most of the protein is on one side or the other of the membrane. An integral protein is a protein that spans the membrane. Like a transport protein has to stick out of the membrane and pump something either into the cell or out of the cell, so it's an integral protein. The cell membrane displays selective permeability, meaning it allows certain molecules to cross and does not allow other molecules to cross. The transport across membranes happens by either simple diffusion, facilitated diffusion, or osmosis. Oxygen can move across the cell membrane by simple diffusion. Fructose can move across the cell membrane by facilitated diffusion. And then water can move across the cell membrane by osmosis. Eukaryotes do have active transport. So if there's a molecule outside the cell and the cell wants it, it will, and it can't move across the membrane, it will put a transport protein on the membrane that, generally speaking, only moves one molecule either into or out of the cell. So, for example, the molecule lactose is a sugar the cell might want if it's in the environment, and the cell can make an active transport protein, put it in the cell membrane, and then bring the lactose molecule inside the cell. Prokaryotes do not have group translocation. They only have active transport. Unlike prokaryotes, eukaryotes have vesicular transport, where transport vesicles can actually move molecules inside the cell from one location to another. And that is a membrane-bound organelle that we call a transport vesicle, transporting molecules from one part of the cell to another. On the plasma membrane, there are two ways to bring something into the cell. Endocytosis can happen by phagocytosis, where pseudopods come out of the cell, surround what the cell wants to bring inside of it, and the pseudopods are just extensions of the cell membrane coming out, and then they can fuse, where the cell membrane will fuse, and then the cytoplasm will then come across. Let me show you that. No idea why sometimes paint comes up and other times it doesn't. When I click there we go. So here's our cell. And then here's something that the cell wants to bring inside. We can build pseudopods right here, right here. Those pseudopods can expand. Further. And then when they touch like that, the plasma membrane can connect. Let me show this with the double membrane. Put this one inside. And then they can fuse. Where this membrane can then connect to that membrane. This membrane can connect to that membrane. Oops, didn't draw that very well. And then this membrane can connect to that membrane. And this membrane can connect to that membrane. Not drawn very well, but you can kind of guess that the cytoplasm is now totally surrounded this. And this is a membrane. There's the bilayer. This, I don't have the bilayer shown then. This is the organelle membrane around what the cell is then brought into the cell. And then this can come move inside the cell. And now we have the organelle surrounded by an organelle membrane and what we brought into the cell. I drew it bigger than there. Any question about any of that? So that's phagocytosis. You can also have something brought into the cell by pinocytosis, where the molecule, let's get paint again. Here's the cell, and a molecule can come down, bind to a receptor. This will then move over here. So this receptor is now moved with the ligand bound to it. So we'll put the thing on it. And then the thing is that the receptors can move together. I'm only going to draw 3. So that's a receptor, and here's a ligand bound to it. And then this can invaginate. So let me draw that in. This can invaginate so that it's now like that, and like that. And then this can invaginate further. That's what's going to happen to the cell. But that's too difficult to draw that again, so I'm going to draw it on the outside. So this brought in further by invaginating further. And then this can be brought into the cell by, once again, forming a vesicle, meaning this will fuse, touch and then fuse. And then it'll form a vesicle here. This will be a phagosome, which will then be brought into the cell. And that's pinocytosis, where the membrane folds inward, bringing in fluid and dissolved substances. Phagocytosis, where pseudopods extend outside the cell and engulf the particle it wants to eat, called cellular eating, and this is called cellular drinking. Materials can move out of the cell by exocytosis, and that happens much in reverse of what I just stated, that a vesicle, like a transport vesicle, can move up to the cell membrane, bringing contents. So this is the cell now, and that's the cell membrane. And the vesicle, which has the contents in it, which we're just going to put molecules inside, it will move up to the cell membrane. Let's get rid of that. Get rid of that. Make this a double membrane. Make this a double membrane. Yeah, there's my stuff inside the vesicle. And then the vesicle membrane can fuse to the cell membrane. So this membrane will fuse to that one. This one is already fused to that one. And then this one will fuse to that one. And this one will fuse to this one. And then the contents here can leave the cell. Any questions about exocytosis? And endocytosis, I guess. Cytoplasmic streaming. The cytoplasm can stream in eukaryote cells. Remember, the cytoplasm is the aqueous substance inside the plasma membrane, but outside the nuclear membrane. Cytosol is just the fluid portion of the cytoplasm. Cytoplasmic streaming, shown here, is where the cytoplasm can move around the cell. Pull it up and forward. There you go. See the streaming where this is moving? These are actually chloroplasts, which are moving around the cell, and it just allows you to see that the cytoplasm is actually moving in this direction here in the cell. Any questions about cytoplasmic streaming? All right. The cytoskeleton is a structural framework of proteins running throughout the cell. It makes the backbone of the cell. The cytoskeleton is made up of three types of proteins: microfilaments, intermediate filaments, and microtubules. You should have had that in Biology 160, actually. Here we're seeing the cytoskeleton. Let me blow this up. So this would be the cell, something like right here. And we're seeing the actin filaments. You don't need to know that. It's one of the cytoskeleton proteins. Here, this would be the cell here. And that's the nucleus of the cell. And this is showing you two cytoskeleton, the actin filaments shown in red and the microtubules in green. So there you can see almost the entire cell here is, what do you call it? Has cytoskeleton running throughout the entire cell. Here on this one here, the this portion of the cell doesn't have the cytoskeleton in it, but all other portions do. Let me remind you that there is a primitive and simple cytoskeleton in prokaryotes. It was discovered about 15 years ago, and for some reason, the textbook author has never updated his book stating that there's no cytoskeleton in prokaryote cells. Ribosomes are the site of protein synthesis. In eukaryote cells, the ribosomes are a little bit larger than a prokaryote ribosome. In eukaryotes, they have an 80S ribosome size. The ribosomes can either be bound to a membrane, meaning the ribosomes are attached to the rough ER. That's why we call it the rough ER, because the ribosomes make this portion of the ER look rough. There can also be free ribosomes. They're just freely found in the cytoplasm. They're not bound to any membrane. Note that eukaryotes do have some ribosomes which are 70S in size. These are the ribosomes found in chloroplasts and mitochondria. Only these eukaryote ribosomes are 70S in size. The ribosomes found in chloroplasts and mitochondria. All other ribosomes are 80S in size. Organelles. An organelle is a specialized membrane-bound structure allowing compartmentalization of functions. Each membrane-bound organelle has a specific shape and performs a specific function in the cell. Here are some different organelles of the cells. The nucleus, the ER, the Golgi complex, lysosomes. You can read about them. I'll talk about the mitochondria and the chloroplast in just a minute. Let me briefly mention the endomembrane system. In eukaryotes, we have an endomembrane system where various organelles are part of the cellular transport system, meaning that the different organelles work together to transport a molecule from one place to another. This is known as the endomembrane system. For example, proteins are made at a ribosome, and if they're made at the rough ER, the protein is made at the rough ER, and they're packaged in a vesicle which forms from the rough ER, meaning the endoplasmic reticulum, puts the proteins in a vesicle, which is a transport vesicle, and then the transport vesicle moves to the Golgi apparatus, fusing with the Golgi apparatus right here. So the vesicle makes the proteins, puts it in a transport vesicle moves to the Golgi apparatus, fuses with the Golgi apparatus, dumping the proteins in this side of the Golgi apparatus, which then sorts the proteins and modifies them, chemically altering the protein. When the protein is all done, it is then move to this side of the Golgi apparatus, and it buds out, making another transport protein, which then transports the protein to where it is needed in the cell, including it could take the transport protein a transport vessel can move to the cell membrane and then dump the contents inside the transport vesicle outside the cell. It can also move them to a certain location in the cell where the transport vesicle then stops moving and it just starts storing the proteins for a certain length of time. And while the transport vesicle is not moving and is just storing, we don't call it a transport vesicle, we call it a vacuole. So a vacuole stores things, a transport vesicle moves things. Any question about any of that? That's the endomembrane system. All right, let's talk about the chloroplast and the mitochondria. I need to talk about the endosymbiotic theory. This is the theory to explain the evolution of mitochondria and chloroplasts in eukaryotes. How did these organelles originate? Well, the theory is a long time ago, before we had eukaryotes, we had a proto-eukaryote, which I'm not going to write up here, just call it a proto-eukaryote. And this proto-eukaryote brought in a cell, we'll try and make it a rod, which could perform aerobic respiration, and it was a bacteria. I've got this capitalized for some reason. If you remember, aerobic respiration can take one glucose and make up to 38 ATP from one glucose molecule. Yep, I got to put in 38 there. The proto-eukaryote could only make ATP from fermentation. So one glucose makes only net gain of 2 ATP. Well, what happened was the proto-eukaryote engulfed this and ate it, and probably just surrounded it by making pseudopods. I don't want to go through that right now. But this was brought into the cell. Actually, let's just change that here. And for whatever reason, the proto-eukaryote did not consume the bacteria that it engulfed right away. Oops, let me say this. And the membrane would fuse, and so this would be brought in a vesicle membrane. So it's brought into the cell with a vesicle membrane. And glucose from the cytoplasm of the proto-eukaryote moved into the bacteria, which could then make ATP. And the bacteria shared some of the ATP, probably because life was early and the glucose left the cytoplasm of the proto-eukaryote and moved into the cytoplasm of the bacteria, which then made ATP, and some of the ATP left the cytoplasm of the bacteria and entered the ATP. We don't know how much ATP was shared by this bacteria, but it was a lot more than 2. So the proto-eukaryote thought, what went on here? I lost 1 ATP, excuse me, 1 glucose, but I gained a whole bunch of ATP. I could only make 2 ATP from 1 molecule of glucose. And somehow the proto-eukaryote knew that this bacteria was responsible for making that excess amount of ATP and using up the 1 glucose. So the proto-eukaryote decided to keep this bacteria, so this bacteria would make ATP from 1 glucose and then share its ATP with the proto-eukaryote. And then both were happy. The bacteria got a home, it got food from glucose from the proto-eukaryote, and then the proto-eukaryote, although it lost 1 glucose, it got a whole bunch of ATP. And then the two started living together for a long time. And now we find this bacteria only inside the proto-eukaryote. And now the proto-eukaryote always has this bacteria in there. And we no longer call it a bacteria, a proto-eukaryote, we call it a eukaryote because it has this cell in it which evolved into the mitochondria. And that's how the thinking came about as to where the mitochondria came from. Any questions about any of that? Anyways, that's the theory. What evidence do we have for the theory? First is that there are some protists, simple protists, which can engulf another cell and then use that cell to make something for the protist. Like this protist has engulfed an algae, which is actually going through division here, and the algae can obtain sunlight and then make glucose for the protist. Furthermore, we do have some, what are those called? We do have some protists which actually lack a mitochondria. And what these protists do, they're amoebas. That's why I was trying to remember the name of it. The amoebas, they will engulf a bacteria that can engage in aerobic respiration. And then the protists will share the glucose with the bacteria. And then the bacteria will use that glucose to make ATP and share some of that ATP with the, forgot the name of it now, the protist. So we actually do have some eukaryotes which do as this theory suggested, although that is a bacteria living inside the protist. It is not a mitochondria, but it's acting as a mitochondria does. Some other evidence when we look at the DNA of a prokaryote cell, it's usually one circular molecule. In eukaryotes, we have linear DNA molecule. The mitochondria and the chloroplast DNA is circular, and there is DNA in the mitochondria and in the chloroplast. Another thing is that the membranes of the mitochondria and the chloroplast, it has 2 membranes. Like on the mitochondria, there is an outer membrane and then an inner membrane. So the inner membrane would be the, let me end the show here. The inner membrane, do I have it still up here? The inner membrane will be the membrane of the bacteria. The outer membrane is the vesicular membrane. So this theory will predict that the organelle will have 2 membranes, which both the mitochondria and the chloroplast do. And they're the only membrane-bound organelles in the eukaryote which have 2 membranes, both an inner and an outer membrane. When we look at the histones in the bacteria, they are lacking in bacteria. No histone proteins. They're present in eukaryote cells. There's no histones on the DNA in the mitochondria and the chloroplast. Furthermore, when we look at the start codon for the first amino acid made in protein synthesis, in a eukaryote cell, it's methionine. In a prokaryote cell, it's not methionine, it's a similar amino acid, formylmethionine. In the mitochondria and the chloroplast, the first amino acid that's made is formylmethionine. When we look at the ribosomes of a eukaryote, they're 80S in size. Of a prokaryote, the ribosomes are 70S in size. When we look at the ribosomes inside the mitochondria and the chloroplast, they are 70S in size. And yes, these membrane-bound organelles, the mitochondria and the chloroplast, have ribosomes inside them. They're the only ribosomes inside a membrane-bound organelle of the eukaryote cell. Furthermore, when we look at the growth of the prokaryote cell, those cells mostly grow by binary fission. Eukaryote cells grow by mitosis, and the mitochondria and the chloroplast grow by binary fission. And then the strongest piece of evidence that the mitochondria and the chloroplast belong to the prokaryotes is when we look at the DNA and sequence the DNA of a eukaryote cell, it falls within the domain eukaryotes. However, when we're looking at the chromosome. However, when we look at the DNA of a prokaryote cell, of a bacteria, it falls within the domain bacteria. When we sequence the DNA of the mitochondria and the chloroplast, the sequence of the DNA of the mitochondria and the chloroplast falls within the domain bacteria. Any question about any of that? All right, so that's the evidence that the endosymbiotic theory came about from bacteria being engulfed by an early proto-eukaryote, which then the two lived together in symbiosis, and now we don't find the mitochondria outside living freely. It's only inside the cell of a eukaryote. And that's true for animal cells. And plant cells, this event happened twice because they engulfed the bacteria, which became the mitochondria, and then they engulfed the bacteria, which became the chloroplast. So in plant cells, it actually happened twice. Any question about any of that? All right, that's the endosymbiotic theory. And that's it for this lesson. Any questions?
I wanted to ask if we have access to these slides, or is this just for your use?
You won't have access to these slides until I've finished with the lesson. Let me take a look and see if the last lesson you have access to the slides.
Maybe I'm just looking in the wrong place. I was looking at the modules during the lab and the lecture portions for the past weeks, and I haven't seen any
Chapter
PowerPoint
2
slides.
is up. Let me show you. Chapter 2, it is published. Let me check Chapter 3. Chapter 3 is up too. I think we just finished Chapter 4, right?
Yes, we just finished chapter 4.
Yeah, I will make Chapter 4 available later this week.
Okay, thank you.
All right. Any other questions? If not, I'll see some of you at 6:30 in the lab. The rest of you I'll see in the lecture on Thursday.
I have a question, but it's about the quiz.
Okay. If it's short, I'll answer it. If it's not short, let's have this question another time.
It's short. It's just one question that was on the quiz.
Uh-huh.
It was looks like it was from chapter 5.
Yeah, I
Um,
got your email. I gotta look that up.
okay, okay. I
That's
thought
why I
maybe
haven't responded. I haven't had time to look it up. So I don't know if you're correct because many topics we talk about in several places. So I got your email. I'm sorry I haven't responded. I just haven't had time to look it up.
okay, okay. Thank you.
Mhm. All right. All right. Bye.